Okay, much of my work has focused on transforming and creating ideas that are from algorithmic thinking to biology. Today, I'll be talking about a way to use machine learning methods from natural language processing to identify ways we can target viruses to develop therapeutics and vaccines. I'm going to put this into slide mode, sorry. So the problem that motivated us is that of viral escape. So when a human is infected by a virus, we make antibodies, shown in blue here, the standard diagram. And these bind to surface proteins on the cell of the virus and are thus recognized by the virus. However, so what these antibodies do is they neutralize the virus or inactivate it so that it can't reproduce. Now with viral escape, what happens is viruses can mutate their surface proteins, and they do so in such a way that they're so substantially different in what we call their semantics or meaning that the antibodies no longer bind to them. And thus, they can escape the immune system, bind to host cells, and cause disease. Now, viral escape is a big problem. In fact, viral escape of influenza, and in fact, through the surface protein, hemagglutinin, of influenza, and the envelope surface protein of HIV, are both highly responsible for the fact that we don't have a universal flu vaccine, nor do we have a vaccine for HIV, both of which cause hundreds of thousands of deaths a year. Now, in the midst of this pandemic, the spike protein on the surface of SARS-CoV-2, the virus that causes COVID-19, we wonder how prone it is to viral escape and whether we should be concerned about this. And there's some evidence already that there is viral escape with this spike protein. So ideally, understanding escape and predicting escape will inform antiviral and vaccine development. And I'm more on the part on understanding escape. So our key idea is to leverage the fact or use the intuition that mutations to a protein can affect the protein in different ways. So for example, here we have our standard picture of a protein, and we're going to use language modeling that originally developed in natural language processing to be able to predict the effects of different mutations on the protein. So for intuition, let's turn to English sentences as an example. So suppose here we have the boy pats the dog. Well, one mutation to the original protein that we're going to model can simply change the protein so that it's not really too different functionally than the original protein. So this would be akin to changing the sentence to the boy pets the dog. It pretty much has exactly the same meaning. And in addition, it's still grammatical. So another mutation to the protein can completely inactivate the virus or cause it to have lack of function. For example, it could no longer fold. And in so doing, it cannot bind to the surface of the cell. And this would be akin in natural language processing to a nonsensical word being inserted into the sentence or lack of grammaticality. So the problem with this mutation, which is different than viral escape, is it affects the action of the protein. And now I'll tell you what viral escape instead does. And so I should mention that affecting the action causes the protein not to obey the biological set of rules or grammar of rules that are put in place naturally. So instead, with viral escape, what happens is the protein is changed enough from the original protein by the mutation so that it is no longer recognized by the antibody. So with the sentence, we could change it to "the boy eats the dog." Now, clearly, this has very different meaning than the original sentence, but it also preserves grammaticality, or the grammar of rules that are intrinsic to the English language. So the problem that we came up with is to look for mutations in the grammar of languages, or the grammar of biology, or the rules of the grammar, that also induce high semantic change. And we call this constrained semantic change search. So in other words, we want enough of a change in the semantics or meaning of the protein, but it still is grammatical. That is, it can still infect host cells and cause disease. So our novel approach is that we constructed a single natural language processing model that simultaneously learns both grammaticality and semantics. Now, this model, is not particularly novel in itself, but before this point, people only used it to get grammaticality or fitness of the virus, or predict that, or they used it to interpret semantics or meaning of the virus for antigenic prediction. So what we do is we do both at the same time. We look for a token that the probability that a token is output by this model based on the sequence context around it. This could be the probability of a word based on the words around it in the sentence, or it could be the probability of an amino acid based on the other amino acids in the protein sequence. And so we interpret the output of the model as an approximation to the grammaticality of the original sequence, and the continuous semantic embedding, which is shown here in yellow in this hidden layer, as an embedding of the original sequence, which allows us to quantify how different the original sequence is from the one that we've input to the model. So this gives us a distance calculation in metric space. So, of course, to give a little more intuition for this, to cause viral escape, it needs to be that we have high grammaticality and we have high semantic change. And that we get out of this model. So as an example, let's suppose we train on newspaper headlines. And the original headline is "The Australian dead in Bali." So the semantically closest headline, according to our model, is "Aussie dead in Bali." And you can see there's not much change in meaning between those, but they're both still correct grammaticality wise. So when we're trying to look for those mutations that are prone to viral escape, our constrained semantic change search model proposes the following change: Australian ballet in Bali. You can see this has a very different meaning from the original headline, but it's still correct grammar-wise. So what we've been able to do with this model, and I want to say that this model will be appearing in NeurIPS 2020, and the reviewers and editor were very excited about its contribution to biology, and it probably wasn't done in this way for natural language processing because there was no reason to preserve grammaticality and semantics. So one thing we can do with this model is that we can take all sorts of sequences that are homologous to the spike protein from a host of species, and we can show their embeddings in UMAP two-dimensional space. So each of these is a sequence, each of the points. And the cool thing is that we can visualize these semantic embeddings. This has nothing to do with grammaticality. It's just what this hidden layer in our model outputs. And we can see that similar sequences, meaning similar semantic sequences, are close to each other. For example, the human MERS-CoV virus is close to camel, where it's zoologically known to originate from. SARS-CoV-1 is close to civet and bat, and SARS-CoV-2, related to the pandemic, is close to bat and pangolin. Another thing we can do is that we can predict escape mutations by not training on those known escape mutations or those verified experimentally. We do not train on those. And that's known as an area called zero-shot learning, which is a really challenging area of machine learning. So here we plotted a mutation to each point in influenza virus hemagglutinin based on our model's predicted score of grammaticality on the x-axis and semantic change on the y-axis. And we have a priority score for our model that goes from darker color to lighter green to yellow. And that's, okay, so, and we color the mutation points based on the model's prediction. So notice that the experimentally validated mutations are shown with red X's. And there's tremendous agreement between the escape predictions that have been experimentally validated and those predicted by our model, which is what we want to see. We also compared our model to state-of-the-art models that also could be used for escape prediction. So the lighter ones are based on, let's say, just looking at the grammar of the model, as I said, or the fitness. And those show that they run on both types of influenza and show these models have these viruses, or the surface proteins have high fitness, and they associate that with high escape potential. The medium gray models, one of which is ours and appealed in ICLR 2020, show that those with high semantic change are associated or somewhat with escape prediction. But if you look at our CSCS model, here shown in dark gray, we see that it is highly predictive of escape potential for these two influenza surface proteins. And similarly, for the envelope protein on the surface of HIV, and for spike, we get much higher prediction of escape for the spike protein in general. And in fact, and we also get it for the receptor binding domain. And I'll go into this a little more on the next slide. So the idea is that we want to use this idea to help develop therapeutics and vaccines. And we want to identify, to do this, target regions that are less prone to escape. And the idea is, if we target those, like people are doing to develop antivirals for flu, then maybe that will apply to all flu strains or all surface proteins on the flu, or on influenza, because we're targeting regions that are less prone to escape, and therefore we might be able to universally target the viral proteins. So here we see that the stalk region has low escape potential, whereas the head region in both influenzas has high escape potential. So in this case, we'd want to target the stalk. And if we look at SARS-CoV-2, the spike protein for that has escape enrichment in the receptor binding domain and escape enrichment in the N-terminal domain, but less so. And the receptor binding domain has high escape enrichment. And curiously, the S2 domain, which is a protein on the surface, is high in escape depletion. So we would suggest that that is the area to target. And the red arrows show where we'd like to target. So the hope is that language modeling can shed light on those parts of a protein that are not prone to escape, and we can target them not only through what's being done for influenza in the targeting of antibodies to regions that don't vary too much, but also in a new field called, really exciting, called immunofocusing, where we develop vaccines such that we train the immune system to respond to those parts of the surface proteins that are known not to undergo escape. I'd like to thank my amazing team of collaborators, Brian He, who pretty much led this work, and Ellen Zhang, both students in my lab, and Brian Bryson, who is the most amazing collaborator. And I'm so glad that we get the chance to work with them. And our paper is under minor revisions at Science. So you'll likely see this published soon. If you'd like to get our software, you can get it at this site. So thank you.
Thank you very much, Bonnie, for this wonderful presentation. It's really funny because I'm moving to biology and you're moving to natural language processing. I guess we're going crosswalks. At any rate, there are a number of questions that people asked about your presentation. I don't think we have time to answer that, to go through all of them, but I would just, would you like to select the questions yourself, or should I select the questions for you?
So I see Q&A here.
Yeah, so there is a first question. How do you segment the virus regions to predict escape potential, for instance?
Oh, how do you segment them? Well, we run on the entire viral sequence. This is, the model trains only on the sequence. It knows nothing about escape potential. And, or we could also segment regions, like known regions, like S2 of, you know, of the spike protein, or the stalk of influenza, or even the stalk of HIV, which kind of brings me full circle because I started my work in computational biology in the early to mid 90s with Peter Kim, and we were able to predict novel coil regions where the head of the protein folded over on the tail, and then by some spring-loaded mechanism, it came off the tail, and the stalk region of the tail permeated the membrane of the viral coat. And we discovered this for lots of viral membrane fusion proteins using our coil prediction programs.
Okay, let's just pick up another question. Are escape predictions the same within all SARS, MERS, CoV-2, etc.?
Well, we did show that slide where our model used the predictions in embedding space, which would just be the semantics and meaning. And we did validate that it was close to others and their similar types. The escape predictions that we've made are that they jive with experimental findings. And in some sense, one point I want to make that I didn't make is that we can do combinatorial predictions with this model. Even though I've only talked about single point predictions, combinatorial are fine. And there's been a paper for HIV that just came out that shows that, not HIV, for SARS, that 4 predictions are known to cause antigenic response of the virus. And we were able to predict all of those. And those are in our paper. And every prediction that we've made and that we say that it's correct, like the X's on that slide, have been validated experimentally. So, it's not like just some simulated thing. We actually have run on all these real data studies, the 2 of the SARS ones that have come out. Now, realize to do this, we need large corpuses. This ties into Regina's sharing. We need large corpuses of sequences and known escape patterns so that we can actually decide when we train our models on the sequences whether our predictions are correct for viral escape. So it'd be really nice that everyone shared their data because we don't have much on escape yet. We don't know much even about SARS. We saw that one study, thank goodness it came out in our last stage of review, but we don't know much.
So, Bonnie, there are lots and lots of questions. If you have time, you're welcome to reply to the answers. We have to move to our new speakers, but thank you so much for excellent presentation. Thank you, Bonnie.
Thank you for inviting me. This has been a joy.
Thank you.